Okay, in this video what we're looking at are carbohydrates as molecules. We're going to look at the structure of carbohydrates and then look at the reactions that are required to hydrolyze or break down carbohydrates through digestion. So just having a quick look at the study design first. The red sections are have been removed to the 2020 study design. So basically cross those out on if you've printed this out, or be mindful that there are sections once again of this topic and this chapter that we won't be looking at and you won't be examined on. Okay, so it's linked. We're linking this, or the reason that these sections are removed is because we haven't talked about what's called a stereoisomer and the glycemic index as well, sort of like another little tacked on section that we don't need to go through. All right, so the main focus, as I said, is all on structure and function. Okay, so just like we did for protein, we're going to look at the overall structure of carbohydrates. And just like protein, they are made up of these smaller units, so proteins or amino acids. For carbohydrates, the smaller repeating units are called monosaccharide. So mono meaning one unit, and saccharide effectively means sugar. Okay, so I'm sure you've heard of glucose and a few of those other sort of, some of you might have heard of sucrose and things like that, and they tend to be used in conjunction with the word sugar and so on. So it's sort of like the more common way to refer to a carbohydrate. So we're going to look at their structures, but these structures you don't really have to remember much of. So I'll go through what's in the data book, but you can see some structures here on this slide. The first thing in terms of overall structure and properties, you can see from this first figure here, okay, that because we've got a large number of the hydroxyl functional groups, your monosaccharides are have really high solubility in water, because each of these hydroxide groups, okay, they're all going to form a hydrogen bond, and even your ether group here, with water, and that increases the solubility dramatically if you've got a large amount of hydrogen bonding occurring. So there are three monosaccharides: glucose, fructose, and galactose. Okay, and each of them are shown down here in this diagram. So you'll see structure, they do look a little bit different. Glucose and galactose are very similar, but what's really important, and you can count up your atoms if you need to, but they all have the molecular formula C6H12O6. Okay, so often when we look at our chemical equations, we will use that as glucose, but technically, in terms of the molecular structure, that could technically be galactose or fructose. It's just glucose tends to be used more because it's more commonly found in the human diet. So they all have the same formula but different structures. Therefore, they are isomers of one another, structural isomers. Okay. Glucose, what's called alpha glucose, is structured like this. So you'll have this 6-carbon ring. So each of these points is where there's a carbon. Sorry, I should say 5 carbons. It's a hex ring with 5 carbons and the 6th one at the top here. The orientation of the hydroxyl groups are really important. So they need to be orientated exactly in that orientation. And for the purpose of the fact that we haven't done stereoisomers and things like that, you'll just have to sort of take my word for it on that. It's really important that they're orientated in that sense. What we haven't included here in these diagrams is the opposing hydrogen groups, okay, which they don't need to be included, and they're not included in the data book pictures either. But just so you are mindful of these sort of half stick, half semi-structured diagrams, what we're actually looking at, each point being a carbon, and the hydrogen is not included on the diagram to simplify the diagram. So the only difference in alpha glucose and alpha galactose is the orientation here of this carbon here, the 5th carbon. The fructose, slightly different structure. So you can see it's got a pent shape. So it's got a 5 shaped ring with a carbon down here and a carbon up there. Okay, so you can quite tell the difference between fructose to the other two. You might be as you might hear or read a little bit about additional isomer of glucose, where the orientation of the first carbon, the hydroxide, is orientated differently. So once again, this I wanted to leave it in because you might hear alpha glucose and beta glucose a little bit. And beta glucose isn't glycogen; it's different orientation here. Oops, and back up of this first carbon. Okay, but it's effectively in the region of, or an explanation for this is more in the region of a stereoisomer and what that actually means, which is not what we're covering. So, but I just wanted to point out, if you do see alpha and beta, that they are technically slightly different molecules. Okay, so in the data book, I've had a look at the recent one, and I'll put this on OneNote for you as well. So we're not technically given every single structure, okay? So you're given alpha glucose, which is good. You're given alpha glucose here, and you're given beta fructose, all right, which is like basically giving you everything. What you need to then know is that, and be able to draw, I suppose, given this diagram, and I've just stuffed this up already, is that your galactose will have that different orientation on that fifth carbon, and everything else will be the same. Okay, so this is your galactose. Once you have all three monomers or monosaccharides, then you can basically determine any structure, a disaccharide, or anything that they want you to draw. The two structures below are a disaccharide, and I'll talk about that in just a second. Okay, so you probably figured it out, but a disaccharide is effectively two monosaccharides bonded together. So it's like a dipeptide, effectively, di meaning two. And there are three versions or three forms of this that we're going to look at. So glucose can bond with glucose itself. So you see that this first example here. And a glucose-glucose bond forms maltose. The bond occurs at the first carbon of one glucose and the fifth carbon of the other. And because the hydroxyl groups are orientated in that kind of downwards position, we show our bond here in that orientation. So that's really important. We show that bonding downwards like that. It is called a glycosidic bond or linkage. I prefer the word bond, glycosidic bond. Technically what it's also called, it's called an ether bond. I'll just write that at ether, which is a type of organic bond we haven't talked about, bonding hydroxyl and hydroxyl together. Okay. A second type of disaccharide called sucrose is made with glucose and fructose. You'll notice the same type of bonding. So bonding with glucose, carbon 1, and fructose, which that would be 1, 2, 3, 4, 5. Yeah, 5 again. Okay. So you see the pattern, just like with proteins. It's a nice and simple pattern. Once you get the hang of it, they're fairly easy to draw. The last one is lactose and glucose, okay? You get a slightly altering bond here due to the orientation of the hydroxyl group of glucose and the hydroxyl group of galactose, and we get what's called lactose. There's actually several different forms of lactose, whether alpha beta, depending on if it's alpha beta, glucose, and so on. So it can get a little bit confusing. Particularly if you try and look up the structure of lactose on your computer. So just have a look here back on the data book. This is the structure of lactose that we've been given. You can clearly see that is a very, very different structure to the maltose one that we went through before. Okay, and this change in bond orientation here is something we'll talk about in terms of how lactose is broken down. All right, so your disaccharides then can form even further into longer chains, and the longer chain carbohydrates are called polysaccharides. So effectively, they're referred to as polymer carbohydrates. Can be very, very long chains of lots and lots of disaccharides effectively bonded together. There are three major types of polysaccharides: glycogen, starch, and cellulose. Glycogen is the form that your body stores glucose. So your body actually can't store glucose in a lot of parts. We actually store excess energy or excess, effectively excess energy as fat, which most people would be aware of. But the body can have a reserve of glucose in the form of glycogen, and it's stored in the liver and in some muscle tissues, but it's certainly not an enormous amount. If you stop eating, you would probably use that up within a half a day or a day, depending on the situation you're in. Starch is effectively one of the most common forms of glucose that you ingest or that you eat, found in a lot of different types of foods. And cellulose as well, but cellulose is a little bit different. Some of you might be aware it is the key structural component of a plant, and therefore when we consume cellulose, we're consuming a lot of plant-based material. This is just a bit of a summary, goes to the monosaccharides, disaccharides, poly. It's a nice little summary table. You might like to mimic something like this in your own summary. But what we need to look at is how we form these polysaccharides. Okay, so the first one I'm going to look at is starch. Now, there's actually two different forms of starch. One's called amylose and one's called amylopectin. But effectively, starch, once again, is formed from the disaccharide maltose, which was formed from the monosaccharide alpha glucose. And that's what you're seeing here. You're seeing these glycosidic bonds occurring, forming your disaccharides, which then continue to bond with one another and form your starch molecule. Okay, that once again, that glycosidic linkage showing that you've got alpha glucose bonded. There is a little section of this, I think, in your data book as well. It sort of shows a repeating units of starch, but we wouldn't be drawing anything more than maybe 4 molecules across and leaving, once again, our bonds open, just like the ends of this one here. Amylopectin is really, really similar, except what actually happens in the formation of amylopectin as well is some of the orientation actually occurs and causes branching. All right, so it's occasional branching roughly every 20 units. So that says that down here. Okay. And we're actually getting orientation with this first carbon and the 6th carbon. So remember, this was a CH2OH, which is forming, it's still once again a glycosidic bond. And that's bonding with the hydroxide of the first carbon. So I'll just flick back to this one here. You can see, so that's what's happening here. You're getting orientation, or you're getting some bonding, some attraction here on that 6th carbon, and that's bonding to the first carbon of a neighboring glucose molecule effectively. Causes branching, changes the overall structure and digestive properties of the polymer itself. So amylopectin is a little bit different in terms of structure, solubility, and so on. If we take a look at cellulose, so cellulose is different once again because it's formed by the polymerization of beta glucose. That's why I talked about beta glucose earlier. So because the orientation of beta glucose on that first carbon means your hydroxyl group was orientating up here at this point here, we get this glycosidic linkage occurring here. So it changes our orientation of our glycosidic linkage. You can sort of see we get orientated down in the first bond, but then, where am I? This one here, orientated down the first bond, orientated upwards in the second bond. Okay, what that actually means in terms of, if you look at the polymer as a whole, it allows for better orientation of the cellulose, the big long polymer strands. So they actually kind of fit alongside each other a little bit better. We get less repulsion and more attraction. And overall what that means for the properties of cellulose is that it's actually a more strong and more rigid polymer than starch. And it obviously works perfectly in plants because cellulose is the structural integrity of a plant. Plants don't have muscle or bone or anything. They use cellulose to maintain that structural integrity. But the bonding here of cellulose is effectively very different from starch. And so what that means is, therefore, to break down cellulose, you're going to need a different enzyme to do so. All right, we can't just use the same enzymes to break down starch as you would cellulose because the bond type here is different. Think about that lock and key model. Even the bonds being even just slightly different, like they are in this picture, means that and the enzyme that we use for starch won't necessarily be able to lock into, or definitely can't lock into, the molecule cellulose. The last type of polysaccharide is glycogen. It's once again alpha glucose, but it's highly branched alpha glucose. Okay, so only difference, very, very similar to amylose, just more branching effectively, repeated every 7 to 11-ish units. Okay, so just a couple of review questions. Pause your video, have a go at questions 1, 2, and 3, and I'll just go through the answers in a sec. Okay, your disaccharide, hopefully you've worked it out. You've used your structures. Don't think you're remembering these structures. They are in the data book. So use the structures and draw them exactly as they are shown. If you don't have the hydrogen shown, that is completely fine. That's just the picture I've got here. You don't need to show them. You need to show the correct monosaccharides, which is glucose and fructose. The name of the molecule is sucrose. The type of bond is a glycosidic bond, and the overall molecular formula is C12H22O11. And what I haven't mentioned, and which you've seen though in the diagrams, once again, it is a hydrolysis reaction. So for every bond, we get a H2O cleaved off. So in your molecular formula, it's C6H12O6 times 2, but then we have minus H, 2 H's and an oxygen off, which is why we get that molecular formula. Another good summary to have a little look at, I'm not going to go through it, but I just thought it was quite a nice little diagrams included just to kind of compare your different polysaccharides. So this is in your hydrolo book. Okay, so the last component is digesting our carbohydrates. And just like with proteins, digestion requires a specific enzyme for each reaction. How it's digested, we start with a polysaccharide. They are broken down into disaccharides, and those are further broken down into mono, and those monosaccharides are able to be absorbed into the bloodstream. All of these are hydrolysis reactions. Okay, so hydrolysis reactions require water. The opposite, a condensation reaction. All right, so hydrolysis of carbohydrates. Let's have a look at an example here. So an enzyme called amylase, you don't have to remember specific enzyme names, breaks starch down into maltose. So it acts upon the glycosidic links between starch molecules and breaks them into maltose. Then a second enzyme is required, and that's because it's a different molecule. Maltose is technically an overall different shape. So always remember, when we talk about enzymes, you've got to remember, think about that specificity due to shape. The shape of the molecule is different. Okay, so maltase is able to then break down maltose into glucose molecules. Okay, and it acts upon this bond here. So it will bind and bond some, to sections of the molecule, and it will help loosen or allow the reaction to break through that bond at a lower activation energy. Once we have glucose broken into its its monosaccharides, glucose is small enough to be able to be absorbed into the bloodstream and used for energy. I'll talk about the energy component at the end. All right, so another example, or just a little bit more detail there, your disaccharide into your polysaccharide into your di, and into your monosaccharides requires water and requires a specific enzyme for each specific reaction. So as I sort of mentioned earlier, I talked a lot about cellulose being structurally different. It is very structurally different to starch, and so what that means is it requires its own enzyme. Cellulose requires cellulase to break it down into its disaccharide and so on. Now, humans don't contain this enzyme. There's a picture of a cow. Cows do. A lot of other animals do, but humans don't. So what that actually means is if we consume cellulose, we actually don't obtain any energy from it or any nutrients. It is used effectively as what we call fiber in the diet. And some of you have done some health and nutrition might know, be able to talk about this in further detail. But what we're looking at more is just structure and function. But it forms what we call bulk fiber in our diet, and it's really important for gut health. All right. But no, when you eat just pure cellulose, you won't necessarily get any further energy or nutrients from that molecule. All right. The last one I want to talk about is lactose. A lot of you have heard of lactose intolerance before. What does that actually mean? Funnily enough, I'm not really sure what the reason is, but for some reason, quite a large number of the human population don't seem to be able to produce the enzyme lactase basically once they get out of infancy. And lactase is required for the hydrolysis of lactose into glucose and galactose. All right, so without that enzyme, once again, we can't break the disaccharide into two. And you can't use maltose because, once again, think about it, this and think about the whole molecule, the 3D structure is very, very different from the structure of maltose. All right, so we can't use the maltase enzyme and just sub it in. It's not going to be able to lock in the active site. It's not going to be able to lock in to the disaccharide and actually be able to weaken that bond and hydrolyze the molecule at all. Okay, so yeah, those people that are lactose intolerant effectively either completely lack or lack able to be able to produce enough lactase enzyme. The last thing is just looking at bringing this all back to why this actually occurs. So remembering that carbohydrates are the main reason. The main reason we eat them is for energy. Okay, so that's the primary function of the consumption of any carbohydrate. Okay, so glucose itself, we break it down into monosaccharides or glucose, galactose, fructose, whichever one you want to talk about, doesn't really matter. They're all the same in terms of chemical formula. They are our primary source of energy. So if your body has one of those monosaccharides, if you have glucose in it, you will use that glucose to produce energy. We've talked about the production of energy in terms of cellular respiration before. On this slide here, I've included several the chemical equation which you've seen before. It's important to go back over that and understand we've got glucose as your reactant requiring oxygen to produce CO2 and water and energy, or what's called ATP, but don't worry too much about that. We won't ever write energy here. Remember, we will always show it as, if we do know delta H value, which it would be a negative value of energy. Otherwise, we would just leave it as a chemical equation. All right, that is the end of this video, which has covered the first two sections of our